Welcome back. So tonight's lesson is actually going to be a pre-lab for the lab that you guys are going to be completing in class tomorrow. So tomorrow's lab is super, super, super important because it is a state mandated lab, which means that questions about this lab will be on your Regents exam in June. So it's super, super important that you guys pay attention to not only this video, but also the lab in class tomorrow, because you will see questions about it again on that Regents exam. Right, so today's aim, because of that, is how can we prepare for the New York State mandated diffusion lab? As always, you have a do now question. So right now I want you guys to pause this video, answer those two questions, and press play when you're ready to go over it. Question number 1 says, in the diagram below, which letter indicates the part of the cell that carries out a function most similar to a function of the human excretory system? So the excretory system's job is to release wastes. And there is an organelle found inside this cell that does something pretty similar to that. And hopefully you got that was B, the cell membrane. The cell membrane, as we know, is the door of the cell. It's going to be what lets materials in or out. Because of that, it's what releases those wastes when it's letting those molecules out of the cell. All right, number 2, which statement best describes the plasma membrane of a living plant cell? Remember, anytime you see the phrase plasma membrane, that means the same exact thing as cell membrane. Okay, so is the answer A, it selectively regulates the passage of substances in and out of the cell. B, it is composed of proteins and carbohydrates only. C, it has the same permeability to all substances found in or out of the cell. Or D, it is a double protein layer with a floating lipid molecules. So the answer here is going to be A. It selectively regulates the passage of substances in or out of the cell. Remember, this selective regulation is the same thing as selective permeability. And when we're talking about selective permeability, we're talking about that certain molecules are allowed to pass into the cell membrane and certain molecules can't. So it is selective in what it regulates or what it allows to come in and out of that cell. All right, so let's get started. Again, this lab is super, super, super important. So make sure you guys follow along with me as we go through this together. Today in lab, we're going to be using, or tomorrow in lab, I should say, we're going to be using something called dialysis tubing. This is used in something that's called dialysis. But what exactly is dialysis? So let's read through this passage together. The kidneys are a pair of organs, each about the size of a fist, located on either side of your spine. They're responsible for purifying your blood by removing wastes and excess fluid from your body. When the kidneys don't work properly, dialysis is used to perform the function of the kidneys. Dialysis is a treatment that filters and purifies the blood using a machine. This helps keep your body in balance when the kidneys can't do your job. Dialysis has been used since the 1940s to treat people with kidney problems. Okay, so dialysis, what is it? Well, if you might, that was not English, let me try that one more time. You might actually know someone who's had dialysis treatment before. This is what happens when someone's undergoing kidney failure or has some sort of kidney issue. They undergo dialysis, so their blood continues to be filtered even though their kidneys don't work. So dialysis tubing is used in that process. So we're actually going to use dialysis tubing today or tomorrow. Now, what exactly is this tubing? So dialysis tubing is a type of semi-permeable membrane. Now, that sounds kind of familiar, but I'm going to keep going. It's a type of semi-permeable membrane tubing used in the separation of molecules from the blood during dialysis. Now, I really feel like I have heard the word semi-permeable before. Maybe with the cell membrane? So you guys have got 3 questions on your paper. The first one is, which organelle is dialysis tubing most similar to? And hopefully you made the connection that this is most similar to the cell membrane. It is semi-permeable, meaning it's going to let some things come in and it's going to prevent other molecules from entering. So it regulates the passage of molecules in and out of the cell. Now you have 2 more questions on your paper. I want you to go ahead and pause this video, answer those 2 questions, and then press play when you're ready to go over them. The second question on your paper says, how does it compare to this organelle? So how is dialysis tubing similar to the cell membrane? Well, you should have said something along these lines. The cell membrane is semi-permeable and it controls what enters and exits the cell. So again, certain molecules are going to be allowed to enter and certain molecules will not be allowed to enter. So how do molecules move across the cell membrane? Well, this week we've been talking about something called diffusion, right? Or passive transport. And in order for that process to happen, the molecules first have to be small enough to fit through that cell membrane. And we know that molecules are going to be moving from high to low concentration. So again, I'm going to write down that this is through diffusion, which is a type of passive transport. All right, so because it is passive, that means that no energy is required for this. And finally, I want you guys to write down, oh no, I've covered up my definition. I want you to write down the definition of diffusion, and that is movement of molecules from high to low concentrations. So again, diffusion is a type of passive transport. We're going from high concentration to low concentration. So remember that slide. We're starting out high, we're sliding down, and we're finishing low. All right. Next, so here is a visual demonstration of what diffusion looks like. Again, we're starting out with a high concentration. Okay, so a high concentration of molecules. And these spaces over here have a low concentration of molecules. And over time, what we see happening is that these molecules are moving down the concentration gradient, which means that they are traveling from high to low concentrations. So the molecules are gradually over time going to move to fill out these spaces until finally we reach this state called dynamic equilibrium. And the word equilibrium kind of sounds like the word equal. And that's because we're talking about equal amounts of molecules spread throughout. So where there once were more and less molecules over here, now there is an equal amount throughout the entire area. All right. Again, this is diffusion. All right, so let's get started. Actually, I lied. Before we get started, I've got one more review for you. I want you guys to think back to Unit 2. We learned about different types of biomolecules and indicators. Well, they're back, so I hope you didn't forget them because we're actually going to be using a few of these biomolecules and indicators in the lab. So what I want you guys to do right now is pause this video again and using the word bank that's provided, fill in the missing blanks in this passage. Some of them will be used more than once. All right, so pause it, don't look at the answers, write down what you know. All right, so here is hopefully what you guys got out of that passage. So I'll read through this. Saccharide is another word for sugar. Starch is a polysaccharide. Glucose is a monosaccharide. Since polysaccharides are long chains of monosaccharides, this makes starch a much bigger molecule. I'm going to highlight that. It's going to come up today. Iodine is an indicator for starch. In its beginning state, its color is amber. And if it's positive for starch, it will have a color change to black. Benedict's is an indicator for glucose. In its beginning state, its color is blue. And if it's positive for glucose, it will have a color change to red, orange, yellow, or green. In order to test for glucose, the solution must first be heated in a test tube. I actually want you guys to read through that passage one more time on your own before we move on to the next part, because each of these concepts will come up in today's or tomorrow's lab. So it's super, super important that you guys know what each of these things mean. Now, here's the time in which I would do a demonstration for you about what you will see in lab tomorrow. But because I am in my home, I don't have any of these lab materials here. I know you're probably thinking, what a horrible scientist. She doesn't have beakers in her home. I'll work on that, guys. So instead of doing this demonstration, I'm going to attempt to explain to you what we would see if I were doing this in class. So if we were doing this in class, I would have a beaker and I would make a cell using that dialysis tubing. So you can see that dialysis tubing right here in the center. It's this thing that looks like a little worm inside the beaker, okay? I would have gotten some of that dialysis tubing. I would have pumped a few squirts of starch and glucose into that cell, and then I would have tied it off with some string. And eventually it would look something like this. So inside that cell, we have starch and we have glucose. And then in this area surrounding the cell, so in my beaker, you might notice that it's a bit of an amber color. And that's because in our beaker, I put iodine. Right, so that's why it's an amber color. I also want you guys to note that the inside of the cell is originally white. Okay. Now, down here at the bottom, I've actually noted the specific amount of molecules that would have been found in or out of the cell. So inside the cell, there's no iodine, but I put 4 molecules of starch and 8 molecules of glucose. And outside the cell, in the beaker, in the solution in the beaker, I put 18 molecules of iodine, and that's it. There is no starch and no glucose in there. And then over time, some time would have passed, and eventually these are the results that we would have seen over here. Right, so let's go ahead and note what we see. Now, this is still our original cell, but my pad's falling. But you may notice that this cell is no longer white. In fact, that cell is now black. Okay, now I want you to think about what may have caused that reaction. So we know that theoretically starch and iodine should still be in there. I'm sorry, starch and glucose should still be in there. But what would have made the inside turn black? I'm going to put a pause on that thought. We'll find out soon. Now, outside the cell, in this solution surrounding it, that color is still amber, which means that iodine is still present. All right, now down at the bottom, we have to fill out the final state of this diagram. So we're trying to figure out, where did these molecules go? Well, what do we know about diffusion? Well, we know that molecules, when they're diffusing, want to go from a high to low concentration until they maintain that equal state called equilibrium. So they're going to travel across that membrane to reach equilibrium, that equal state, equal amounts of molecules. Okay, so theoretically, these molecules of glucose inside the cell are going to want to travel across the cell membrane until they reach equilibrium. Same thing for this starch. Starch is going to want to travel across the cell membrane to reach equilibrium. But what do we know about the size of starch and glucose molecules? So while glucose is a small molecule, starch is actually a really big molecule. Remember, it's a polysaccharide. And because of that, starch is actually too big to cross the cell membrane. I'm going to put a frowny face. It can't cross the cell membrane. So because of that, starch cannot diffuse across the cell membrane. Right? But the same thing is going to happen here with iodine. Iodine, because there are 18 molecules outside, it's going to want to diffuse into the cell again to reach equilibrium. So let's go through what theoretically our final results should be. So while originally we started with 0 molecules of iodine inside the cell and 18 outside of the cell, they're going to eventually reach 9 and 9. So we'll have 9 molecules of iodine inside the cell and 9 molecules of iodine outside the cell. Again, they're going to diffuse into the cell because they want to achieve equilibrium. They want to have the same amount of molecules on the inside as they do on the outside. The same thing's going to happen here with glucose. While originally we had 8 inside the cell, those are going to diffuse out of the cell this time. So we'll have 4 molecules of glucose inside the cell and 4 molecules of glucose outside of the cell. So again, equilibrium has been met. There are even amounts on the inside, even amounts on the outside. But here's our sad little friend starch. Starch was too big to cross the membrane. So while iodine and glucose were able to reach equilibrium on both sides of the membrane, starch couldn't escape the cell. So in the ending result, we're still going to have 4 molecules of starch inside the cell and 0 molecules of starch outside the cell, which is kind of sad. He didn't get invited to that party. He couldn't make it. All right, so let's go back to why was this color black? This ending color of our cell was black because iodine diffused across the membrane. Because iodine is an indicator for starch, it turns black in the presence of starch. And the reason that our ending color of our solution was still amber is because there is no starch in that ending solution. Right? So in our ending result, outside the cell, we had iodine and glucose. And inside the cell, we had starch, glucose, and iodine. And because iodine met starch, the ending color here was black. All right. If you have any questions about this, please let me know on Remind. We will be doing a longer version of this in class tomorrow, where hopefully your questions will be answered. The last thing we're going to do today is write out these results. So what happened? How can we write this down? So what happened to iodine in this experiment? Well, iodine is small enough to diffuse through that membrane. Okay, so iodine was small enough to travel across the membrane. Because of this, it diffused and turned the starch inside a black color. All right, what happened to starch in this experiment? Well, starch, remember, he wasn't invited to that party. The starch was too big to diffuse through the selectively permeable membrane. So it did not diffuse out into the surrounding solution. And finally, what happened to glucose in this experiment? Well, glucose was small enough to fit through the selectively permeable membrane. This indicates that glucose diffused into the surrounding solution. And we'll do part C tomorrow, but when we test that outside solution for glucose using Benedict solution, remember that's the indicator for glucose, after heating it, the solution will turn a variation of red, orange, yellow, or green. Now there is one more question number 4, that I want you to answer on your own before you turn this in. I want you guys to state one reason why a molecule may not be able to pass into or out of a cell. So think about what we just saw in this lesson, and I want you guys to tell me, what is one reason why a molecule may not be able to pass in or out of a cell? There it is. Oh, that's ugly. There you go. What is one reason why a molecule may not be able to pass in or out of a cell? All right, so make sure you answer that question before you press submit. As always, let me know if you have any questions, but please turn in both this and your exit ticket before tomorrow morning at 8:30. And I will see you guys tomorrow. Have a wonderful day.